Hello friends, welcome to another video tutorial from Akash Biology. In this video, we will continue our videos on the phytohormones. So in this particular video, we are dealing about cytokinin, which is a type of a growth accelerator hormones. And talking about to the same pattern, first of all, we will be talking about the history of how cytokinin was discovered. So, how cytokinin was discovered? So in 1954, in the year 1954, Skoog and Miller Skoog and Miller was experimenting on the tissue cultures, mainly on the autoclaving of the disrupted DNA of herring fish sperm. So they were during the process of tissue culture, they were autoclaving the disrupted DNA that was obtained from herring fish sperms, and they found out that materials contains something which has a very good role in increasing the cell division activity in the tobacco pith cultures. So they obtained a material that increases cell division in callus of tobacco pith culture. Tobacco pith culture. And as it was a growth accelerating form, so they named it as kinetin. They named it as kinetin. So kinetin was the first cytokinin to be discovered. But as it was discovered from a tissue culture form, so it is the synthetic form. Synthetic form or the artificial form. So it is the synthetic cytokinin. Synthetic cytokinin. So the first cytokine that was discovered was not natural. It was a synthetic form, and it was named as kinetin. Now coming on to the another things, after the Skoog and Miller, there was a scientist known as the Latham. So in the year 1963, so Latham obtained a similar materials that was same as that of the kinetin from unripe maize kernels. Unripe maize kernels. As the scientific name of maize is G-maize, so he named it as zeatin. The zeatin. So zeatin is the first natural cytokinin. First natural cytokinin. So we are mainly being confused in the examinations. Which one is the following first cytokinin to be discovered? So first cytokinin to be discovered is kinetin, but it was a synthetic form, as it was discovered from or being processed from a tissue culture form. Later on, the first natural cytokinin was discovered from unripe maize kernels by the Latham, and he named it as zeatin. So zeatin is the first natural cytokinins. So coming on to the precursor form, so from what of the following substance does the cytokinin is being formed? So it is formed from a purine. Purine. So aminopurine. Aminopurine. So as it is a aminopurine means amino means base. So it is a basic hormone. Basic hormone. So unlike gibberellin and the auxin, it is not acidic. It is a basic hormone. And the precursor element is the purine adenosine. Adenosine. Simple. So adenosine is the precursor, and it is a aminopurine, and it is a basic hormones. Now after the cytokine is being discovered, now there were huge demand of the cytokinin, because they want to accelerate the production by increasing the cell activity. Cell divisions. So the scientists were busy in discovering the synthetic forms which may be available in short period of times. Though now there are a lot of synthetic cytokinins in this particular scenario. So the the synthetic forms are so ethyl, let me review the names, I have forgotten. They are ethyl adenine. These all are synthetic forms. Similarly, there is benzyl adenine. Third one is the benzyl aminopurine. So there are lots of cytokinins, synthetic cytokinins, but these are three prototypes that you need to mug up for the entrance examinations. Now coming on to the parts of the plant from where we can get the cytokinins. So cytokinins are present in all of the parts of the plant where there is active cell division ongoing. So they may be in the tip of roots or the tip of shoots or in the buds or in the callus or in the growing leaves. So any of the place wherever there is what increasing amount of cell activity or there is activity of the cell division ongoing, then we can find out what there is a cytokinin. Now coming on to the what, and it can also be found in the coconut milk. So you may be asked in the exam famous questions, it is found in coconut milk, apple juice, and yeast extract. What? Yeast extract. So these are the things that you need to mug up. And similarly, fourth is the growing endosperm of plant. And the unripe maize kernels, do you know? This is a simpler thing. So these are the main points that you need to mug up from the entrance point of view, from where we can extract the what cytokinin. Now coming on to the various bioassays. So I have told you earlier what each bioassay. Bioassay is the proof of that substance is cytokinin by applying it to the living system and assessing its functions. So there are lots of bioassays. So first of all, bioassay is the retention of chlorophyll. Now bioassay. So first is the retention of chlorophyll. What? Retention of chlorophyll. And these all bioassays are being performed on knowing the function of that very substance. Second is gain of weight in tissue culture. As it is found in the what endosperm, so radish, exist, cotyledon size assessment. So in all of the what we have to take a test solution. What? Test solutions. Another one is what? Control. So control means the natural conditions. So we will compare. So all of the bioassays are being performed on comparing the two substances. In the test we have sprinkled or we have dissolved cytokinin. So this is what? Cytokinin. In this there is a natural environment. So if you are going to be a doctor, you need to know about the test and control. So any of the experiment is being performed or hypothesis is being tested for an experiment. So we have to take a test solutions or a test environment in which that particular substances is being sprinkled or dissolved, and a control in which there is assessment in the natural environment. So first of all, coming on to the retention of chlorophyll. So it is often known as what? It is based on what? Richmond-Lang effect. It is based on Richmond-Lang effect. So what we will do? We will take a leaf. So leaf is undergoing senescence, means the leaf is undergoing aging process. So aging natural phenomenon. So we have taken a leaf. Now the leaf is being excised into the two parts. What? The leaf is excised into the two parts. Now one of the leaf is tested in what? The test solutions, and one is put into the control solutions. Now after 72 hours we will see the leaf. So the leaf which is in the natural condition has undergone the process of aging very fast. But the leaf that is being placed in the cytokinin environment has a retardant process, appears longer as it is. So that very phenomenon is known as what? Richmond-Lang effect. So Richmond and Lang found out that in 1980s that when a leaf is being sprinkled or the leaf is placed in an environment of cytokinin, then it retards the process of senescence or the natural aging, because in the cell it increases the invertase activity. What? It increases the invertase activity. So this is one of the very important thing that you need to know. So Richmond-Lang effect is being tested for a cytokinin in the leaves, and it due to the increase in activity of invertase. Similarly, in what? Gain of weight in the tissue culture. Gain of weight in the tissue culture means we will take a tobacco pith culture. Now it is being divided into two parts, one in the test, another in the control. So which one is placed in the test solutions will weight more after 3 or 5 days, and which in the control have less weight in compared to the test solutions. Similarly, the radius cotyledon has to be departed into the two sizes. One is put into the test, another in the control. So test मा जुन जान्छ त्यो ठुलो हुन्छ, कन्ट्रोलमा सानो हुन्छ. टकिङ इन्टु द नेपाली हामी रिचमन्ड ल्याङ इफेक्ट भनेको के हो? नर्मली लिफहरू पनि के जान्छ, नेचुरल फेनोमेना अफ एजिङमा जान्छ. त्यो एजिङलाई रोक्नु पर्ने या फिर फ्लावरहरूलाई हेर्छौँ धेरै दिनसम्म हामीलाई के राख्नुपर्छ, फ्रेस राख्नुपर्छ. त्यति बेला हामी के गर्यो त, फ्लावरमा साइटोकाइनिन स्प्रिङ्कल गर्यो भने त्यसको सेल वालमा के बढ्छ, इन्भर्टेज एक्टिभिटी, र यसको नर्मल जुन फेनोमेना एजिङ छ त्यो प्रोसेस रिटार्ड हुन्छ. त्यसैले हामी के भन्छौँ त, रिचमन्ड ल्याङ इफेक्ट, जुन इक्जाममा एकदमै बढी सोधिएको छ, इन्भर्टेज एक्टिभिटी. सिमिलरली गेन अफ वेट इन द टिस्यु कल्चर. सो एउटालाई राख्ने टेस्टमा, अर्कोलाई राख्ने कन्ट्रोलमा. त्यो टेस्टले बडी वेट गर्छ, यसले कम गर्छ, किनभने सेल डिभिजन एक्टिभिटी बढाएछ. त्यस्तै रेडियस एक्सिस्ट कोटिलिजन साइन्समा पनि हो. Now we will going on to the physiological effects of cytokinin. What the physiological effects of cytokinin? So coming on to the physiological effects. So physiological effects, first is the what? Cell growth. Cell growth means it increases the size of a cell. What increases size of a cells? Second one is cell division. So it increases the, or accelerates the process of cell division. Third one is morphogenesis. What morphogenesis? So you need to know about the thing from the entrance point of view. So if we sprinkle a solutions which has high cytokinin to oxygen ratio, that it will increase what? Shoot. Differentiation or the morphogenesis of shoot. Similarly, low cytokinin by oxygen means the differentiation of root or the morphogenesis of root. Similarly, intermediate cytokinin by oxygen means differentiation of both shoot and root. And lastly, if there is intermediate cytokinin and low oxygen, then it will increase the morphogenesis of callus. Then you can copy it by pausing the screen. So this is about the morphogenesis. Now similarly, prevention of senescence. What? Prevention of senescence means the prevention of senescence. So it is same as the Richmond Lang effect. Fourth one is what? Increases shelf life of plants and vegetables. When the vegetables and plants are cut, same as flowers, and if I sprinkle cytokinin over it, then it will not become, it will remain fresh for a longer period of time. Similarly, it increases the flowering in short day plants. What? Short day plants, like as in the lemna. 6 is the what? It increases femaleness in those plant, femaleness in male plant, and the another thing that it increases the phloem transport. What? It increases the phloem transport. So phloem transport means the phloem are present. What? Similarly, if we see any root, so xylem is conducting water from the root to the shoot. Now food is being formed in the leaf, so the food will transport from what? The leaves to the root. So what? The phloem transport is being increased by the process of cytokinin. So if we see let's say this is my hand is a flower. So this is what? The apical root. This is what? The lateral buds. So whenever there is a food is being transported to each and every part, so the lateral buds again grow. So it will suppress what? The dormancy. It will suppress dormancy is being decreased. Similarly, the apical dominance is also decreased. Apical dominance is decreased. किनभने हामीले खाना सबैमा पुगाउने lateral budsले पनि same as that of substance पायो in the same manner as that of the apical buds. त्यही भएर apical dominance पनि के हुन्छ? जान्छ. अर्को भनेको एउटा मात्र बच्यो अब के हामीले एप्पल प्रोडक्सन बढाउन लागि. So if you want to increase the product size of apple, then we sprinkle it with what? Pomalin. What? Pomalin. So pomalin is what? Cytokinin plus gibberellin. What? Gibberellin. So gibberellin में क्या होता है? GA4 and GA7 and cytokinin में क्या होता है? 6-benzyl adenine cytokinin. So this is the pomalin, and it increases apple size. What? Apple size. So this video is all about the cytokinin. I hope you like the video, and if there is any of the feedbacks that you want to give me, then you can give me on my YouTube channels, and you can also follow me on Facebook, that is on Akash Biology, where you can have access to all of my videos. And this is a channel in which we are providing the topics on demand, so you can write any of the topic in which you want my next lectures. And in this month my theory exam is ongoing, so I'll be posting the videos so late, so my apology in advance to you, because I will not be posting the videos very regularly, because I have to also prepare for my exams, though I will try to reach you as soon as possible. And stay tuned and connected with Akash Biology. And please, my humble request to everyone, if my videos are helpful to you, then please like and tap the subscribe buttons and share it in your Facebook, so that everyone can get access to my channels and knowledge can be spread to everyone.